And welcome and thank you for joining us today for the next of our Stanford 22q11 Seminar Series. Today, our topic is Segmental Duplications and 22q11 Deletion Syndrome. So first, I'd like to introduce Dr. Alex Urban, Professor of Genetics and Psychiatry and Behavioral Sciences at Stanford, who will be moderating our seminar today and introducing our speakers. So thank you, Alex.
Thank you, Rachel. Am I on, or did you want to do some housekeeping?
Just our housekeeping is just we'll have our Q&As after each speaker speaks, and so during their talk, please feel free to post your questions into the chat, and we'll deal with them at the Q&A. Thanks.
Okay, fantastic. Well, oh, Bo, go ahead.
I have a technical thing. It seems like there's something going on with the wireless in the Porter Drive building. If the Zoom keeps shutting off and the Wi-Fi goes off, so if that happens again, then I'll move to another building.
Yeah. Maybe you. How about you email your slides to Rachel?
I have them already
then
to
she
go
could.
as
oh, you
well.
have them? Okay.
Yeah.
And then you could call in. You could call in and of course, then you won't see the slides then, but maybe you can synchronize this. Then. Yeah, you can see them. Okay. Yeah. All right. Okay. So welcome again, and I'll try to keep this short because we have such interesting talks, tightly packed. So welcome again, and thank you, Rachel, first also for organizing this, both for very competently running the Hamilton Neuropsychiatry program, and for organizing this seminar series, and in the process also putting up with me being less than helpful in organizing. So thank you very much. Also thanks to the Hamilton Foundation.
Under the house is
I'll come and help you in a moment. Sorry, I have a personal genetics experiment here. So about the speakers, we have three fantastic speakers today. We have Dr. Beverly Emanuel, who is the Upham Endowed Chair in Pediatric Medicine and Professor of Genetics at the University of Pennsylvania and the Children's Hospital there. We have Dr. Zhou, who after a stint as a postdoc and research scientist in my lab was just recently promoted to instructor of psychiatry here at Stanford. And we have Lisanne Vervoort, who is listed as a PhD researcher, but she actually recently defended successfully her PhD thesis at the Catholic University in Leuven. So it's now Dr. Lisanne Vervoort, who is, I think, briefly still a postdoc in Joris Vermeer's lab there. And then in a few weeks or a small number of months, she will be moving to Boston to do her postdoc work there. And sorry about this. I'm broadcasting from home. No, you really need to be quiet now. Huh? Okay, sorry, I'll keep, okay, I will mute in a moment. Yeah. I'll keep it really short then. I apologize for this. The timing is a little bit off. These three researchers, actually, for me, symbolize kind of the frame of my own work on 22q11, because when I, about 20 years ago, as an early graduate student, decided to try and take on this important and really challenging problem, or to participate in taking it on. Of course, I immediately became aware of Beverly Emanuel's work, who had been laying the foundation for everything we're building on today. And also, when I contacted her, she immediately sent me the first 10 or so DNA samples from 22q11 patients, and then we analyzed. We worked on those together. And she walked me through it, really. And then fast forward to today, then a few years ago, Zhou joined my lab, and after I had told him of this challenge, and then also thanks to the Etiology Foundation, who both funded the work that he'll be talking about, and then hosted a seminar at Stanford where I got to talking to Joris Vermeer, and then the collaboration between Stanford and Leuven started, and you'll hear all about that. So I'll be muted now, and please go ahead with the series of talks. And thank you very much. And sorry for the little disturbance here.
Okay. Okay, so I will start the conversation talking about the low copy repeats on chromosome 22. What we know for a long time now is that the genomic architecture of this region, which is commonly deleted, is pretty complicated in that there are blocks of low copy repeats, 8 that in particular, and the ones in green are ones that in fact are responsible for abnormalities that we see. And in fact, the 1122 translocation, which is a recurrent balanced translocation in humans, the most common. Numerous other translocations take place at low copy repeat B and within that region. The most frequent microdeletion syndrome, which is what we will be focusing on today, and shown in the brackets are the low copy repeats A to D, which give you the 3 megabase deletion, which is the most common. And I will talk a little further about that. And the bisatellited marker chromosome that is seen in association with cat eye syndrome. Early on, when we were part of the Genome Center for Human Chromosome 22, using BACs and COSMIDs and PACs, we were able to put together what we thought was an organization of the LCRs in 22q. And I don't expect anyone to quickly memorize what you see here, but what's notable is that in common colors are elements that are common and repeated in the low copy repeats, and so they're color coded. But if you look at the arrows beneath the individual ABCD low copy repeats, you see that in addition to resembling one another, they also change in orientation. So the LCRs are modular in structure, they range in size, and you'll hear more about this 160 KB unit. They contain previously identified markers. At the time, they were markers from the Human Genome Project, etc. Truncated genes, a few coding genes, link RNAs or long interspersed non-coding RNAs, and pseudogenes. And as I said, there's variation in the content number, orientation, and organization of these modules, and they are the sites of many chromosomal rearrangements. But the one that we will talk about today is the 22q deletion syndrome. It previously went or goes by many names: the George syndrome, velocardiofacial syndrome, and in the Japanese literature, conotruncal anomaly face syndrome. The estimated frequency is somewhere 1 in 2,000 to 4,000 live births, and it segregates as an autosomal dominant disorder. There are numerous physical findings, cleft or palatal anomalies, but not in every person. So about 69%, congenital heart defects of the conotruncal characteristics. So interrupted aortic arch, tetralogy of Fallot, and in addition, learning disabilities, speech and language abnormalities. And notable are the psychiatric abnormalities, where schizophrenia usually is 1 in 100 or 1% in the general population. These individuals run a risk of somewhere between 25 and 30% of developing schizophrenia in adolescence or early adulthood. Immune dysfunctions and mild facial dysmorphia. And I like to show this patient who was the first one that I ever had contact with that I know of, who was an unbalanced 1022 translocation and got us sort of in the realm of chromosome 22. These are several of our patients with the deletion. On the top are the youngsters, the infants. On the bottom are 2 teenagers. They have characteristic faces, which include mid-facial flattening, hypoplastic alanase, hooding of the eyelids, cup-shaped ears, and a long philtrum. And what you see is they look sometimes more alike to one another. They are not related than siblings might. And so we were interested in the LCRs and the deletion mechanism. Normally, the LCRs ABCD would line up and pair during meiosis, but if you have a crossover at LCRD, you might get shuffling of some of the elements, but nothing important. However, there is the possibility of misalignment followed by non-allelic recombination. And so if A and D happen to pair and a crossover takes place between them, you wind up, if you follow the top line across, with a duplication on A, and ultimately if you follow the bottom, from bottom to top, you wind up with a deletion on homolog B, so a case of 22q deletion syndrome. And if you look at the track of this region in the UCSC genome browser, you can see these low copy repeats going up to 98 or 90, greater than 99% similarity. And so there are A to D deletions in 85%, A to C in roughly 3%, A to B in a smaller number, and then the translocations. So these deletions result from non-allelic homologous recombination. They're due to the low copy repeats, which we represent as A, B, C, and D. They flank the region and lead to these aberrant exchanges of the chromosomes that result in either a duplication or a deletion. And I must say, as you will hear, finding the mechanism and the breakpoints has been somewhat elusive. This slide was before the most recent T2T sequence was published, where the reference sequence contained gaps on chromosome 22. The new sequence actually closes some of those gaps, but it's a very simple and a haploid genome. So it's a simple LCR and haploid genome. We knew over many years that resequencing failed to resolve this region on 22, the LCRs, because the sequence reads would basically pile up, which led to problems with this particular disease or multiple diseases, and looking at the evolutionary analysis of 22q11.2, which remained for a long time very much unresolved. And as I said, we know about the non-allelic homologous recombination, but except for several exceptions, the recombining sequences and the breakpoints were not found. So we decided that this would require an orthologous approach to traditional sequence-based methods and arrays. And so we decided to take on optical mapping to analyze the LCRs. And so we applied the BioNano labeling system, which uses a DLE1 enzyme, which is a methyltransferase. It fluorescently will label very long DNA molecules, we hope often greater than a megabase long, at CTT AAG motifs. Then the molecules are drawn into nanochannels and held stationary while they are automatically imaged using a multicolor BioNano genomics instrument. And so what I'm showing at the bottom is the HG38 reference with the DLE motifs noted. And doing the BioNano, you get many, many long molecules, which are then aligned into contigs. And some of the sites obviously align very well and are in dark green, other sites not so well. And this was a collaboration between my lab and Ming Zhao's lab several years ago, when Stephen Pastor was then a graduate student, and he is now no longer a student and running a great deal of the bioinformatics optical mapping. We decided to map trios, in other words, two normal parents and an affected child. So the cells are grown, long DNA molecules are extracted, they are labeled with the enzyme, linearized, and then imaged. And then what has to happen is consensus maps need to be organized based on the presence of labels. And then looking at the trios, we would determine parent of deletion origin using polymorphic markers in the parents and the proband to determine which markers were on the normal allele in the proband, which meant the other parent was the parent of origin. And then we would look at the LCRA region, basically lining it up using DNA that is unique to get started, and then look at LCRD. And then in the middle is where the overlap is, or the non-allelic homologous recombination site. So why did we use trios? We actually wanted to understand the organization of the region, which had defied understanding for quite some time. We wanted to determine why it's predisposed to NAR. Are there any predisposing or mitigating configurations of the low copy repeats? Does the position of the breakpoint alter the phenotype? And that is sort of future goals as we move forward. And we wanted to know whether population differences in deletion prevalence are based on differences in structure. And a question that is one that still, I think, plagues many of us was, do the low copy repeats recombine and create new structures during meiosis? Because as I will show you, the configuration is quite complex. So for LCRA, the features that we looked at to be defined by optical mapping were that yellow SD22-3 unit, which is shown at the very top, and the SD22-4 160 kb unit. And these were originally named by Demerol et al. And actually the 160 kb came from Bernice Morrow's lab. And what we did to look at LCR ABCD, we would anchor in unique sequence 5' of the initial LCR, and then anchor in 3' unique sequence at the 3' end, and then take a look at different haplotypes based on whether the yellow element is present, absent, or inverted relative to the reference sequence. And the same is true of the 160 kb, whether it's present, absent, inverted, or copy number variable. And in addition, there are label differences that become essentially polymorphisms, and we were able to sort haplotypes based on these multiple features. And so by the time we finished looking at 88 parental genomes, what we found, if you look at the left-hand side, there were 61 differences based on presence or absence of labels. And on the right-hand side, the integers on the right, there were 32 different basic organizations of presence, absence, or orientation of the 160 kb unit and the gold SD22-3 unit. So that's a significant number of differences in LCRA, really based primarily on just the first genomes that we looked at in parents. We then looked at LCRD, which is a lot less complex, again, anchoring in unique sequence and focusing on the presence or absence of this SD22-460 KB unit and this 64 KB pink unit, which is frequently inverted relative to what is in the reference sequence. And the structures across parental genomes are not quite as complex. Based on label polymorphisms, there are 17 different units, and based on the presence or absence of the red or the pink elements, there were 5 different configurations. But when we were somewhat disappointed when we looked at parents of deletion origin to see whether there was anything in these differences. And basically, the parents of deletion origin versus the non-transmitting parent were fairly similar in whether the pink element was in the reference orientation or in the reverse orientation. And we focused on those elements because when we looked at the probands, there were, it was obvious that most of the recombinations occurred between a forward-facing red 160 KB unit in LCRA and LCRD, with the LCRD pink element, the 64 KB duplication, in the reverse orientation. And then we were fortunate enough to study a very unusual family. The father carries the 1122 balanced recurrent translocation, which he inherited from his mother. His sibling, and this is a family that I had studied quite a while ago, has the supernumerary derivative 22 as a result of the translocation, now known as Emanuel syndrome. He himself, the carrier, has 2 children. One is normal and one has the 22q11.2 deletion syndrome. We thought that the deletion would likely be maternal in origin, and we were surprised that the parent of origin studies determined that the deletion had occurred in the father, and the unaffected child inherited the father's normal 22, and the deletion in the affected child was a deletion between LCRA of the derivative 22 and LCRD of the normal 22. So usually with a balanced translocation, we know from basic biology or what we learned long ago, there's a quadrivalent that forms during meiosis. And one could imagine that the recombination takes place in the region between the paired derivative 22 and the normal chromosome 22 by just a slight shift in pairing during the quadrivalent. By doing the optical mapping haplotypes, we saw that the proband, in fact, has the deleted chromosome that has part of the father's homolog number 1 and part of the derivative 22 homolog number 3. But the recombination actually takes place within, based on polymorphisms, within this particular region of the 160 KB unit of the father. And the sibling has the paternal normal homolog 1 and has the less complicated maternal homolog number 2, where the proband actually has the complicated configuration. And this, describing this family has just recently been accepted for publication. So what have we learned so far? Certainly the LCRs show unprecedented size and structural variability. The LCRA alleles range from 250 kb, the smaller one, to over 2,000 kilobases. There is really no explicit whole haplotype predisposing configuration that has been identified. All the NAR events contain a segdup containing or a module in the segmental duplications containing FAM230 gene members. This actually suggests some preferred recombination sequences. These units, in fact, have palindromic AT-rich repeats around them. The NAR events occur between FAM230 and, as I already mentioned, specific orientations of segmental duplications within LCRA and LCRD. So reference orientation 160 kb modules in A and D, flanked by an inverted 64 kb in LCRD. And the one thing that I will say is that so far, all of the trios that we have examined are either white or Hispanic. And what Donna McDonald-McGinn had noted, and we've since confirmed, that there seems to be a paucity of African-American individuals in our cohort, which is quite large at Children's Hospital. If we looked at a 100-mile radius in our cohort, 16% of the sample are African-American, where 68% are whites of European descent. And if we look at our hospital demographic of the same region, 34.9% of CHOP patients are African-Americans and 51% are white. So the difference between 16% and 34% has a p-value that is 2.2 times 10 to the minus 16, suggesting there is clearly a difference. And so we thought, is it because of features? Do they have less of the phenotypic features? And basically answer is no. In previous studies done, the prevalence of congenital cardiac defects is similar in African Americans as white. So that's actually the most likely reason that children come to attention at CHOP. And our question was, are there factors in the LCRs, in the architecture, that make them less permissive to non-allelic homologous recombination? So we looked at LCRAs in 50 normal African Americans, and there are differences in LCRA as far as copy number and variation. There were 29 LCRA haplotypes unique to African Americans. African Americans have 4 unique haplotypes that have not been seen in any other population, where 18 haplotypes are unique to whites. And few of the African American unique haplotypes have been seen in white parents of deletion origin. And we know that LCRA haplotypes are shared between the parents of deletion origin and white non-deleting parents. But what about LCRDs in the African Americans? That 64 kb LCRD inversion has a lower prevalence in African Americans. And there are many more unique haplotypes in African Americans, primarily in the form of truncated 160 kb duplications at either the 5' or 3' end. And there are structures in African Americans in LCRD which have not yet been found in whites. And if you actually look at the bottom here at the forward pink element versus the reverse pink element, the difference in African Americans is striking in terms of the presence or prevalence of the inverted version of that 64 kb element. Again, this is early information. We need to do a lot more studying. So conclusions, there are clearly differences in the structure of the LCRs between African Americans and whites. And maybe these differences account for differences in prevalence of the deletion syndrome in African Americans. And obviously we need to study additional trios of deletion families to be certain that this finding is significant. So finally, we know that this region is extremely complex and hypervariable in structure. They appear to exhibit genomic instability, which creates susceptibility for not only the deletion, but several other types of chromosomal rearrangements. And it's unclear whether the LCR structure is subject to change by allelic recombination that adds or deletes modules and leads to the very complex LCRA structure. And this is a topic that we will be looking at in large pedigrees segregating through multiple generations. So far, we have not seen that. And we don't know how the LCRs affect or alter the prevalence of disorders in different populations. Right now, African Americans versus whites. Obviously, we need to study different populations and different rearrangements. And we certainly don't know how the LCRs affect different anomalies, say the duplications or cat-eye syndrome. And we've been thinking about this as an exploration of 22q11.2 as a prototype for other complex regions of the genome. And so I need to acknowledge a huge team. Obviously, I get to talk about what many, many people have done. This is a group of part of our team at a lunch, but I really have to thank our CHOP clinical team and the lab team, Stephen Pastor, Mike Z, who's actually over here, a number of technical assistants over the past, but particularly Wen Tran, who has done a ton, and our collaborators, Ming Zhao, Bernice Morrow, Huyan Kwok, and the 22 Deletion Syndrome Consortium, and certainly the families as well. So thank you for your attention, and I'll take any questions.
Thank you so much, Dr. Emanuel. That was just really fantastic. I think we've got time for a few questions, and there are quite a few in the chat here. So firstly, from Alex Urban, you mentioned evolutionary implications of the structure of the 22q11 region. Could you expand on that a bit? And also a related question from Nikolai von Ers. Since the LCRs are restricted to humans, not sure about ape species, is there a possible evolutionary benefit to retain the LCRs within 22q11.2?
Okay, so let's see, getting to the ape story, I know that Joris's lab, as well as others, have looked, they are at, the LCRs actually are in some of the apes. So I would defer to the people who have studied that. It's kind of hard to say whether this gives us any kind of selective advantage. The real question of the evolution is how did they evolve to be as complex as they are, and how long has that evolution been ongoing, and what will we learn as we begin to look at other, perhaps even earlier, more ancient populations, if those sequences become available.
Yeah, thank you very much. And I think we'll move on to our next speaker now. Dr. Zhou, And I think, Bo, would you like me to share your slides, or do you want to try from your computer first?
I can try to share from my computer.
Great.
Okay. Hi. So my name is Bo. I am in Zhou. I am in the Department of Psychiatry in Alexander Urban's lab at the Stanford University School of Medicine. So thank you so much for your attention. So today I'll be giving a talk called Resolving the Exact Sequence Rearrangements and Breakpoints of the Typical 22q11.2 Deletion using CRISPR-targeted ultra-long read sequencing. So the reason why we're interested in, So the reason why we're interested in 22q in particular is its role in the genetic architecture of psychiatric disorders. So we see that usually a range of psychiatric disorders are highly heritable across the human population. And if we look at the various genetic components that contribute to psychiatric disorders, we see that CNVs, or copy number variations in particular, by far contribute the most, confer the highest risk and the highest penetrance. So of these group of CNVs that confer very high penetrance for the development of psychiatric disorders, the 22q11 deletion is at the very top. So the 22q11 deletion is one of the most common microdeletion syndromes in the human population, and the typical form is de novo, and it deletes about 3 megabases of the human genome, and it's also one of the highest known risks for the developing schizophrenia. So here we are really asking some very fundamental questions about the deletion. So namely, where exactly do the DNA breaks occur? What exact sequences are missing and remaining after the deletion occurs? So why is this so challenging to address these two questions in particular? So as mentioned earlier, so the formation of the typical 22q11 deletion, it forms between the homologous recombination between two very large low copy repeat regions. So they, as was mentioned earlier, they range from 250 kb to up to 2 megabases for the LCRA haplotype. So these two highly repetitive and homologous regions of the human genome, so LCR22A and LCR22D, they come together and recombine, and that leads to a deletion. And so in the span of the deletion, 3 megabases of the genome is deleted, and also a cohort of genes in the middle. So now, why is it so difficult to map the exactly where the deletion occurs and where the breakpoints occur? So here I am showing a coverage map of the alignment of short next generation sequencing reads on the 22q11 region. So here we have a female that's diagnosed with schizophrenia. On the top track in the blue, we show all the reads, sequencing reads that map to this 22q11 locus. So from how the reads pile up, we can clearly see a heterozygous deletion on the 22q11 region. And then this is with all the reads that are sequenced. Now, if we look at only the reads that can be mapped uniquely, or unique alignments, namely that we are 100% confident that where the reads map are exactly where the reads come from, then we can see that reads from the LCR22A and LCR22D regions drop out because they are so repetitive, and we cannot be certain which reads actually map to these regions. So that's why it's very difficult to determine where the breaks occur using traditional sequencing methods, adding to the complexity. So even though whole genome sequencing is now routine and talked about a lot, but large fractions of the genome are still inaccessible to standard sequencing analysis, namely centromeres, telomeres, and segmental duplications, or LCRs. And in addition, that for the 22q11 LCR22A region in particular, there are still in the HG38 reference genome, at least there are still 3 large reference gaps that are over 100 kb in length. So
telling us that even the reference genome for this region is now very well put together or even misassembled. So as mentioned earlier, also to even add to the complexity, the LCR22A region. So this is a paper that's published in Joris Vermeersch's lab in genome research. So the LCR22, where they used fiberfish and optical DNA mapping approach, where they map the haplotype structures of the LCR22A and D regions. So what's striking is that the LCR22A is highly polymorphic across different individuals, in the sense that there were a total of 26 distinct haplotypes that were discovered across 22 different individuals. And I think all individuals are heterozygous for this region. So you can see it's very highly polymorphic across individuals. So that explains why the HG38 reference genome is not well put together, as there's still many gaps in the LCR22A region, because you cannot collapse this diversity of haplotypes into a single reference genome. So to address these two questions, the strategy we use is to isolate the deletion haplotype intact on the 22q11 region from the rest of the genome and then decipher its sequence. So to do this, we developed a experimental technique with this is done with Gi-1 Xin from Han Liji's lab at Stanford. So where we use, we combine CRISPR targeting and pulse gel electrophoresis and Oxford long read sequencing to address this question. So I'll go through how this experiment is set up. So here I'm showing a diagram of the 22q11.2 region. On one of the haplotypes harbors the 22q11 deletion, and the other haplotype is the non-rearranged normal haplotype, as this deletion is heterozygous. So we designed CRISPR guide RNAs that cut the outside of LCR22A and LCR22D. And so because there are two haplotypes, the same CRISPR guides will also cut on the normal haplotype. But due to the large size difference between the haplotype harboring the deletion and the normal haplotype, these two haplotypes will separate during gel electrophoresis, allowing us to capture just the deletion, the haplotype with the deletion. So in a little bit more detail, so first we start with patient cells that have 22q11 deletion. These cells are put into an agarose cassette where the lysis happens inside the agarose. And then after cell lysis, genomic DNA is then immobilized to the side of the agarose gel cassette. Here we perform CRISPR guide digestion. So where the cutting happens after the digestion is complete, then we perform pulse gel electrophoresis that separates the two haplotypes, or in more particular, the 22q11 deletion from the rest of the genome, and then it gets eluted into an elution module. We capture that DNA and then perform Oxford nanopore sequencing, and then we can get N50 up to 65 kb using this strategy. And afterwards, we then perform a qPCR on the eluted DNA to verify that we did indeed capture the haplotype with the deletion and nothing in the normal haplotype. So here we have qPCR probes targeting different regions of 22q11. So one probe flanking the LCR22A region, one probe targeting the deletion region, and another probe targeting the LCR22D region. And then we can see in this experiment, at least, that only probes that target the LCR22A and D region are enriched, and nothing in the deletion region, showing us that we did indeed capture the LCR22, sorry, 22q11 deletion. So after that, we then perform Oxford nanopore sequencing, and then we align the reads to the genome, and then we verify that the reads are indeed aligned to where the CRISPR guides cut. And from there, from those anchoring regions, we then perform sequencing assembly, where we take reads above 30 kb, and then we can assemble the entire 22q11 deletion at base pair resolution. So this is the first pilot experiment that we did for a individual with 22q deletion. And here are the reads. showing where they map. So they all map to the LCR22A and D regions. And this is the sequencing assembly of this particular deletion. And for this particular sample, this was also orthogonally validated using fiberfish, where the experiment was done by Lisanne from Joris Vermeer's lab in KU Leuven, Belgium. And she'll be the next speaker. And so now, after describing the strategy that we used to map the deletion, so I will talk about the strategy we used to map the 22q11 breakpoints and deciphering the mechanism of CMV formation. So we have a few families where we have both the parent of origin and also the patient with the 22q11 deletion. So this is also done with Lisanne when she was visiting Stanford during her PhD. So using the same strategy, the CRISPR CTLR-Seq or CRISPR targeted ultra long read sequencing, we can actually isolate the haplotypes from the parent of origin, namely the LCR22A and 22D haplotypes that contributed to the deletion. And with knowing this, what the sequence is prior to de novo CMV formation, and then we can do direct sequence comparison to map exactly where the breakpoint occurs. So using the same strategy, we can isolate LCR22A and D from the parents, and then also the deletion haplotype from the patient. So here I want to mention that when using this strategy, because there's no size difference between the two LCR22 or LCR22D haplotypes, we are capturing two haplotypes. So there will be a phasing and deconvolution step in this process. So after capturing the LCR22D, we perform this phasing deconvolution to decouple the two, or actually no, to assemble two haplotypes for LCR22D, and then determine exactly which haplotype contributed to the deletion. And then we perform direct sequence comparison to map exactly where the breakpoint occurs. So using this strategy, we were able to do this for a few families. And for at least in these 4 families, we found that the breakpoints all occur within the GGT2 pseudogene region. Some occur intronically, some are in exons. But what's interesting is that they all seem to map within retrotransposon regions. So 2 of them map within allo regions, one of them in ERV and the other in an L2 region. So suggesting that this recombination might be a retrotransposon mediated recombination as the mechanism. So in summary, we developed a generalizable method, CTLR-Seq, to solve the exact sequence of large neuropsychiatric CMVs. So using CTLR-Seq, we solve the breakpoint, the sequence and breakpoints of the typical 22q11 deletion, and mapping the breakpoints of the typical 22q11 deletion showed variability in breakpoint locations across different patients, and they also map to retrotransposon elements. And without happy to take any questions. Thank you very much. Oh,
thank you so much. And I think what we'll perfect, thank you. I think what we'll do is we'll move on to Lisanne, and we'll take questions together at the end if we have time. Thank you so much, and Lisanne, we look forward to your talk.
Okay, hello everyone. Can you see my screen now?
Yes, that's great. Thanks.
Okay, perfect. Okay, so yeah, here in Europe, it's already good evening, everyone. So I'm Lisanne Vervoort. I'm a PhD in the Laboratory for Cytogenetics and Genome Research at the KU Leuven, in the laboratory from Professor Joris Vermeers. And during my PhD, I was focusing on identification of recombination hotspots in the 22q11.2 deletion syndrome. So as Professor Emanuel already explained, the deletion syndrome is the most common genomic disorder in humans, and it's characterized by a high interpatient phenotypic variability. And our research actually starts from this feature. So there is this interpatient variability, which is not well explained, or we don't know why this is. And then in addition, the low copy repeats in general are important important in human adaptation, and it's also known that they have structural variation in the human population. But low copy repeats as well are known to be neglected in standard analysis pipelines. So therefore, our hypothesis was that these structures on chromosome 22 could explain part of the phenotypic variability in the 22q11.2 deletion syndrome. So what is the problem with these low copy repeats? Well, actually, as you see, this is a little bit a schematic representation of the blocks A until D, and they're all compiled of different blocks, different subunits, which are present several times in the same block, and then also in other blocks on the same chromosome. So this means that when you have a read from short or from standard long read sequencing data, that there are multiple mapping options for this specific read. And this will result in alignment errors, in assembly errors, and even in the presence of 3 sequence gaps in human reference genome HE38. So therefore, during my PhD, it was the aim to develop and design methodologies to assemble these LCRs and then use these methodologies for population scale studies and evolutionary studies, and then also in the end for the identification of rearrangement sites. So first of all, I will show you the methodology. So Professor Emanuel already explained the bio-nano optical mapping technique. And here in Leuven, we were mainly focusing on another complementary technique, which is the fiberfish. And in the fiberfish is actually a targeted approach to have a look at these low copy repeats. It starts with a combing of very long DNA molecules on a glass slide. And then this slide with the DNA on it is hybridized with specific LCR targeting probes. After scanning, you can manually indicate regions of interest, and these regions of interest are then used for the de novo assembly, where haplotypes are compiled based on matching colors and distances between the probes. And then afterwards, we could actually decompose all of these haplotypes to a duplicon structure. So we use this in the context of a population and evolutionary study. So here I first show you the results of our population study, which were performed in collaboration with the team of Philadelphia. And so, as Professor Emanuel already explained, we found an extreme level of variability within this LCRA haplotype. So we investigated 44 alleles with the fiberfish, and we found that there were more than 25 haplotypes present with sizes ranging between 250 kb until over 2 megabases. In addition, we also performed an evolutionary study on the great apes, and there we found that the chimpanzee, for example, or all the chimpanzees that we investigated, that there was no difference in their haplotypes, so there is no interspecies variability, and then also that this is a very short and minimal haplotype. This could indicate that this expansion and variability that we observed in human is also very human specific. It can be explained by random drift, but maybe there is a human adaptation potential for this locus. Then, as a next step, we wanted to have a look at the rearrangement sites in the patients. So for that, we could also use our fiberfish. What you see here is actually a family, so where we have a parent with a normal LCRA and a normal LCRD allele. And then we also have a patient with an LCRAD rearrangement. We can see that proximal in the haplotype from both the patient and the parental A, everything is the same. So LCRA in the parent is the same as LCRAD in the patient. Then we have this red box, and this red box is actually sequence or subunits that are shared between these three alleles. What we see then is in the patient, distal from this red block, we don't have the green subunit as expected from LCRA in the parent, but we have the magenta subunit which is from LCRD in the parent. So this means that the crossover should have taken place within this red box. The problem here is that this red box has a length of 160 kb, which is still very long to go through with sequencing. So therefore, we used ultra-long read sequencing in our laboratory from Nanopore Oxford Technologies. And the main advantage here is that we have N50 values, which are like median read lengths between 70 and 120 kb, which is very high. And so therefore, we were able to identify the breakpoints in some cases. And I will show you now 2 examples. In the first example, we have a parent and a patient with an LCR-AC deletion. This means between blocks A and C. So here you can see the schematic representation of the fiberfish probes. So also here you see that in the beginning everything's LCR-A in the patient, and then in this shared locus or distal from this shared locus, it switches towards LCR-C. So we performed long read sequencing, ultra-long read sequencing of both the parent and the patient. And this was followed by de novo assembly of the parental LCR-A, the parental LCR-C, and then also the patient AC allele. For this, we use Novolosi, which is a haplotype-specific assembler, which can differentiate between the 2 LCR alleles, and it was developed in our lab. We used then the shared sequence between these 3 alleles for a sequence alignment of this 15 kb locus. And this was followed by SNP identification of LCR-A and LCR-C specific SNPs. So an LCR-A specific SNP in the LCR-AC patient rearrangement allele is when the SNP is present in the parental A, in the patient AC, but not in the parental C. And then LCR-C specific SNPs are the other way around. We plot these SNPs along the assembly position of the patient, and then we can indicate a locus where there is a switch from LCR-A towards LCR-C specific SNPs. And if we have a closer look at this locus, we see that the rearrangement took place in an ALU element. Then the second example is an LCR-AP deletion. And here, if we compare the patients to the parental alleles, we see that we cannot indicate a specific locus or a specific locus where the rearrangement took place, but that is very clear delineation between these 2 B2 probes. So in the patient allele, the first 3 probes belong to LCR-A of the parent, and the last 2 belong to LCR-B of the parents. So therefore, we were mainly interested in the sequence that is present between these B2 probes. So also here we did whole genome ultra-long read sequencing of the parents and the patient. And we see that in the patient, between these 2 B2 probes, there are a lot of repeat elements. There are palindromic AT which repeats, there are Lisanne elements, there are satellites, and so on. And then if we compare this to the parental alleles, we see that in the beginning of the recombination allele, everything is corresponding to LCRA from the parents. And then it switches towards LCRB from the parent. And the point where this switch occurs is a transition point. And this is located in a palindromic AT-rich repeat. This is actually very interesting because palindromic AT-rich repeats or patterns are known to be involved in the rearrangement mechanism of translocations on 22q. They can form hairpin and cruciform structures and in that way increase the instability of the locus. This was described also by the team of Professor Beverly Emanuel. However, they were never reported to be involved in the rearrangement mechanism of deletions as well. So to wrap up this project of the breakpoint identification in the patients, so first of all, we did this fiberfish on 23 patient parent duos, and we could indicate a region within the LCRs where this recombination took place. Already here, we could see that there is variability within the different LCRs on the locus where the recombination took place. However, we really wanted to go at nucleotide level, and for that we used nanopore ultra-long read sequencing approaches on 8 patient parent duos. And so here we see that a few of them had the involvement of the sign and line repeat elements, and this could indicate that non-allelic homologous recombination is the important mechanism. Then we also have some cases where palindromic AT-rich repeats seems to be important in the rearrangement mechanism. And so based on the translocation research on 22q, this could indicate that maybe non-homologous end joining is acting upon these low copper repeats as well. And then in addition, we also performed sequencing and fiberfish on atypical deletions. And atypical deletions means that one of the breakpoints is not located in a low copy repeat. Based on the breakpoint sequencing, we identified microhomology at the breakpoint junctions. And so here we assume that replication-based mechanisms are the basis for these atypical deletions. So we see that there is variability of the rearrangement locus, but also variability of the involved mechanism. And so this variability of the structure of the LCRs, of the rearrangement sites, and of these mechanisms could maybe indicate why or explain why the 22q11.2 deletion syndrome is a most frequent genomic disorder. For the future perspectives, it will be very interesting and necessary to have a look at genotype-phenotype correlations, and then more specifically to see what is the effect of the LCR variability on the transcriptome level, at the 3D level, and so on. And then also, if we have a look at the parents, to see if there are any alleles predisposing to recombination and to the 22q11.2 deletion. So here I'm at the end of my talk, so I want to thank you all for your attention, and if there are any questions, I'll be happy to take them.
Thank you so much, Lisanne. And I think we will, for those of you who can stay on a little bit longer, we'll take some questions for Lisanne and Bo. And we've got two in the chat already from Alex Urban. So first I'll do for Dr. Bozu. The retrotransposon fragments you saw at the breakpoints, were they fragmented? And if yes, down to how long, or is it too early to tell?
The L2 fragments is definitely fragmented. The ALUs are, I don't remember. It's probably too early to tell. Yeah.
Thank you. And Lisanne, oh, actually, Alex is here. I don't know if you wanted to ask yourself, Alex. Okay, so fiberfish and bionano are both fantastic methods. Would there be a synergistic benefit if one were to apply both to one and the same sample? or can you just use one or the other?
Yeah, we have validated that, so together with the team from Professor Emanuel, that are actually complementary, so that they can give the same result for an haplotype. However, with BioNano, it's also possible to, for example, in a specific duplicon, to have a better look at variation within this duplicon, and so to add a little bit more information there.
Great, thank you.
If one had to choose, one would going forward use BioNano, or is there also a benefit to fiberfish that BioNano can't offer?
Well, the benefit from BioNano is, of course, that you do a whole genome. So if you're interested in other loci, it's also very easy to have a look at these. I think for fiberfish, the DNA lengths are a little bit longer. So in some cases where with BioNano, they are not able to compile the complete haplotype, we were able to do it with fiberfish. So I think that's the main difference. But then we are talking about the very, very long alleles.
Okay, that makes great. So ideally one has both methods at hand. That one is covered. Okay, thanks. I've been meaning to ask you that after your PhD defense and forgot. So now I get that question in. Thanks.
Fantastic. Well, with that, if there are no more questions, I just want to thank all of our speakers again, Dr. Beverly, Emanuel, Zhou, and Lisanne Vervoort. And thank you all for attending. We've recorded it, so we'll be putting it on our web pages soon. And thank you all again.
Thank you.
Bye, everyone.
Bye.
Thank you. Bye.